So when we left off with part 1 of chapter 8, we were discussing mitosis, that general cell division, asexual reproduction, and the steps of mitosis: PMAT, prophase, metaphase, anaphase, telophase. So we'd made it through prophase, the first phase of mitosis. When the DNA was condensing, we're starting to see these spindle fibers form. The centrioles are going to each side. We didn't see any sort of order at all yet in the chromosomes. The second phase, metaphase, we're starting to see order. Metaphase, think middle. The chromosomes align on the middle line of the spindle fibers. So they're all right along this line. Metaphase, middle. And then during anaphase, the chromosomes at the centromeres get pulled, one to each side. So in anaphase, you'll see this movement to each side. They look like they're getting dragged by the centromeres because they are. I like to think of that one as anaphase. The A over here and the A over there are separated by the N. So the chromosomes are getting separated. And then telophase, you've got the chromosomes separated to each new cell. So here's going to be one daughter cell, here's the second. And then you're going to have the DNA go back into a nucleus and start to string out again and not be like chromosomes anymore. You also have cytokinesis occurring, where you can see the other organelles are getting split to each side, and then you've got this cleavage furrow forming. So P-M-A-T. And here's cytokinesis. There's the cleavage furrow forming, separating out these two new cells. So cytokinesis will start at the end of anaphase and go through telophase. There's another view of the cytokinesis. You'll have this ring around the middle that contracts to separate them. So it starts just before telophase at the end of anaphase and divides the cytoplasm, the fluids and organelles throughout the rest of the cell. It is going to be different in plants and animal cells. We're going to look a little bit different. What we've been looking at here is animal cells. I think it's actually easier to see the parts in plant cells because they've got that nice cell wall. So here you can see they take little membrane-bound sacs of cell wall and they line it up on the middle. That's called the cell plate. And after that's fully formed, you'll get the new cell wall. So what I like to do with my students is I like to give them examples of plants as well. So this is what's called an onion root. That's the part of the onion that's dividing rapidly. It's growing. So in the area of rapid growth, you'll see a lot of different cells in different phases or stages of mitosis. What happens when you diet is you freeze those cells in their different portions of division or non-division. So each square is a cell. So this cell, for example, this cell, we can see a dark spot. That's the nucleoli where RNA is being formed for the ribosomes. But we can't really see any chromosomes. Whereas right here, for example, we can see chromosomes, but they're still kind of in a nucleus, and they don't have any order yet. So that would be a prophase. And remember, it's not like it goes P-M-A-T. It's a smooth transition between them. So you can see an early prophase versus a later prophase, where the cells are going to start moving around. So I would have you pause the video and try to identify in each of the squares, as well as with the donuts, what phase each of those is in. So pause, go through them, and then we'll double check it together. Okay, A is in metaphase. Those chromosomes are lined up at the middle of the cell. B, that would be probably late anaphase. You can still see they're getting pulled to each side. Right here, or right here would be a little earlier anaphase, where they're still a little bit closer to the middle but getting pulled to each side. That's a really nice anaphase as well. And then C, that would be telophase. So we're starting to not be able to see individual chromosomes. They're going back into a circle. And that cell disc, the new cell wall, is forming here. And D would be prophase. You can see the chromosomes, but they're not in any sort of order yet. Interphase, 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 prophase. You can't see chromosomes there, but you can here. And over here, interphase, non-division, you can't see chromosomes. Here, prophase, you can see the chromosomes, but they're not in order. The nuclear envelope, that membrane is dissolving. Metaphase, they're at the middle. Anaphase, they're getting pulled to each side. Telophase, you're starting to reform the nucleus. Chromosomes are getting a little less visible. And you've also got cleavage occurring here, the cytokinesis, until you've got two new cells. So normally our cells have a control system that tells them when they need to divide and when they don't. If that's some part of their cell division system gets out of control, that's called cancer. So normally cells in an area, if they're close to another cell, it'll tell them not to divide. But in the case of cancer, that stop sequence has been turned off, so now those cells divide rapidly into other areas. And those areas of growth, the masses, are called tumors. If the tumor stays where it originated, it's called benign. Benign is nice. It means it's not spreading. If, however, it spreads to other areas, that's called metastasis, spreading. That would be a malignant tumor. Malignant is bad. Mal is bad. If something is malodorous, it smells bad. Malignant tumors can spread. And how they usually spread is through your lymphatic system. If someone has a malignant tumor that's spreading, that's what we call cancer. So for example, breast cancer, what that will generally do will go through our lymph vessels and spread to other areas like our lymph nodes. Our lymph system, it goes around and collects the fluid and proteins from our blood and returns them back into our blood flow. So then it's also important for filtering. It's part of our immune system to help make sure nothing gets out of hand. But if our immune system is overwhelmed, these cancer cells can travel through the lymph tissue into other areas. So how do we treat cancer? We want to disrupt cell division. Because our cancer cells are going to be going through division at higher rates than anywhere else, if we can target them, they won't divide, and our immune system can go through and take care of the cells. Because our immune system does recognize that there's something off about these cells. They have some sort of altered state. So you can do radiation therapy. So for example, here we've got prostate cancer, and they're putting in these transmitters, and then they put an x-ray over, and then they hit the targets of those transmitters for radiation. And then chemotherapy, that's use of chemicals. So here you've got chemotherapy drugs going in, causing cells to be unable to divide, or they can kill cells. There are behaviors that can change your risk of getting cancer. Now, once someone gets cancer, it's not very helpful to look at maybe if they'd done something else, they wouldn't get it. But you can be doing things right now to be healthier and decrease your risk of getting cancer. For example, not smoking. Lung cancer, generally 80% to 90% of people who get lung cancer were smokers. Adequate exercise, avoiding exposure to the sun, a healthy diet, performing self-exams, and visiting doctors to get tumors identified early. If you can identify them when they're smaller, before they've spread, you have a much better chance of survival. It is important to think about in our society what you can do to make sure that you're healthier and that you're around to see everything that you want to see. So we looked at mitosis, asexual cell division and cell growth. Now let's look at meiosis, sexual reproduction. Why would we ever want to do sexual reproduction? You have to go find another mate, you have to make sure they're like good genes, and then you're losing a percent of your genetics that you're passing on. To each kid, you only pass on half of your genetics. So where's the benefit? Well, the benefit is that during fertilization you get variable traits. So here, these are two twins, they're non-identical, from the same parents. And just for skin color, you can see that they have a variable trait there. She got more light colored traits, genes, and she got more versions of alleles of genes for dark skin. This is important when our environment is less stable. So remember, I said asexual. If your environment's stable, unchanging, you want to do asexual reproduction if you can. If your environment is unstable, if there's lots of parasites or viruses, then you want variation in offspring, because some offspring are going to be better suited for that environment. Do you know what's going to happen in the environment? No, but that's where natural selection comes in. And hopefully some of your offspring will be able to make it in that new environment. So what we have to do is we have to cut the amount of genes we're sending on in half. So like you've got 46 chromosomes, 23 pairs. Where you got that was you got one set from mom and one set from dad. And that's in all your normal body cells, the somatic, your normal cells. And all people of the same species, all individuals, have the same numbers and types of chromosomes. So everybody has two sets of chromosomes 1, two sets of chromosome 2, for example. You will see slight variation, but we'll look at those special cases a little bit later. Okay, so you've got, for each chromosome, like chromosome 1, you've got a copy from your mom, a copy from your dad. When the cell divides, they make a second copy. Those are the sister chromatids. When you look at both copies of your chromosome 1, one from mom and one from dad, those are called homologous chromosomes. Homologous, homo, same. So they're the same chromosome, chromosome number 1. They have all the same genes. So like they code for the same things. They code for your eye color, for your skin color, for your blood type. But they can have different versions. So one side could say you get A blood, the other could say you get B blood, for example. So remember, we've got 22 normal body pairs, and then the sex chromosomes, X and Y. 22 autosomes, those are our normal self chromosomes, autoself. So what we do, we have to go through meiosis. So as normal body cells, we've got 46 chromosomes. We've got what's called 2N, diploid. We've got two copies of each, two copies of chromosome 1, etc., like we looked at already. So what happens when we go through meiosis is we go down to only one copy of each. So we might only get mom's chromosome 1, dad's chromosome 2, dad's chromosome 3, for example. And then those combine together, the egg and the sperm that each have one copy, to make this fertilized new organism that's diploid. It's got the two copies of each chromosome again. And then through mitosis, we go to our regular adult's form. So we are diploid. We've got the two sets of chromosomes where our gametes, our sex cells, are haploid. They only have one. So throughout meiosis, our goal is to get down to only one copy of each chromosome. Then our gametes, the sperm and egg, fuse together, and they form a new cell called a zygote. And then that will start going through divisions to become new life. So here's our normal body cell. This is chromosome 1, mom's copy, dad's copy. Before we even go through any sort of division, we duplicate during the S phase of interphase to make it two copies of each. So what would normally happen in mitosis is one copy of the blue would go to each, one copy of the red would go to each. Instead, in meiosis 1, we just send one copy of each chromosome, one set of chromosome 1